Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas, and in today's class I am going to describe this model of the journal embryology to you, and this model contains so many models in this one model, so we will describe all of these one by one. First of all, we will start from this stage. So here you can see, in this stage you can see that this is the mature oocyte, the secondary oocyte, which has started its second meiotic division, but its second meiotic division is arrested, and it will complete its second meiotic division only in the case when the sperm will arrive. So this is the cytoplasm, this one is the nucleus, and here you can see this layer is zona pellucida, which is not clearly visible in this model, and outer to zona pellucida the cells which are present, these cells are forming the corona radiata. Next, in this stage also you can see that the nucleus which is present over here, and after the when the sperm touches the oocyte, it's zona pellucida, the signals stimulates the nucleus, the cells, and this cell starts to complete its second meiotic division, and it will form the vesicular nucleus. So here in this slide you can see the vesicular nucleus which is present, and this is the cytoplasm of the oocyte, and outer to this you can see the zona pellucida. At this stage you can see the both the male and the female pronucleus are present, and this area is the perivitelline space, then the zona pellucida, and both the nucleus which are present. Next to this you can see both male and female pronucleus then become the vesicular nucleus. So we cannot differentiate between the two vesicular nucleus, whether it is female or male pronucleus. The mitotic spindles appear between these two, and the cells, the nuclear chromosomes comes to lie at the metaphyseal plate, and then the fertilization take place. So here in this stage you can see the two nuclei which have been formed after the process of the fertilization. So these are the different stages. First we have seen the model in which the oocyte is present. Here the oocyte has started to complete its second meiotic division. Here both nucleus, from the male and the female, both are present. Both nucleus has developed into the vesicular form, and then the first both nucleus present after the metaphyseal plate. Here they are present at metaphyseal plate, and then two nuclei which are now been formed, and then they will divide the nuclear membrane and cell membrane, and two cell stage will arrive. So here you can see this is the initial stage when the male and the female pronuclei are present. Here you can see the two cell stage embryo is present. Here you can see the four cell stage embryo is present. Here you can see the cross section of the four cell stage embryo. So the four different cells are present, and here the complete four cell stage. Next stage is, just a minute. So here in this stage you can see over here that first of all we have to differentiate between the amniotic and the chorionic cavity. So as we know that the allantois is an outpouching of the chorionic cavity. So here allantois is outpouching of the yolk sac. So here you can appreciate the allantois which is present. So this cavity which is present, this is the definitive yolk sac, also called the umbilical vesicle, and this is the amniotic cavity. This layer in pink color is the epiblast. This layer in yellow color is the hypoblast. The amniotic cavity, the definitive umbilical vesicle, and definitive yolk sac or umbilical vesicle, the allantois which is present over here. So on this side you can see this gray color tissue is the extra embryonic mesoderm, and the extra embryonic mesoderm which is lining the chorionic cavity, which is lining the yolk sac, is called the splanchnic layer, and the extra embryonic mesoderm which is lining the amniotic cavity as well as the inner aspect of the cytotrophoblast, this is called the somatic layer of the extra embryonic mesoderm. Outer to this somatic layer, here you can see also that this gray color area is the somatic layer, then this one is the cellular layer, the cytotrophoblast, and then the syncytial trophoblast, and then the inner cell space. These are the maternal blood vessels which are present over here. Next to this stage, here you can see in this model, the process of gastrulation has been taken place, and here you can see the folding of the embryo along with the neural fold which is forming the head tube. This is the amniotic cavity which is present over here. This area is the cardiogenic area. This green color is the notocard which is present. This area which is present over here, this is the primitive foregut which will be incorporated into the body of the embryo. Here you can see neuroenteric canal is present, as well as the primitive streak is present. This is the definitive yolk sac which is present, and this is the connecting stalk where the extraembryonic mesoderm is present, and also this extraembryonic in the extraembryonic mesoderm you can see the blood vessels which have developed, and both the cytotrophoblast and the syncytial trophoblast. The next stage after this is this one. So here you can see, just a minute. Okay, now here in this stage you can see that this is the embryo. This hole is the embryo, and this is the head fold of the embryo. This area is the area of developing heart. Here you can see the brachial arches and pouches which are developing. This is the area of the septum transversum. Here you can see the umbilical vesicle which is present. Here you can see the chorionic cavity. This all is chorionic cavity, and this all is the amniotic cavity. Here you can see in this area, these are the basal plate where the chorion frondosum is present and forming the placenta. So this chorionic plate, the basal plate, the primary or the main villi or the stem villi, the free villi or the floating villus, the intervillus spaces which are present over here. This one is the layer where the chorionic leaf is present. So here the minimum development of the chorionic tissue, while over here the maximum development of the chorionic tissue which is forming the chorion frondosum. So this is the development of the placenta. So again I repeat, the umbilical vesicle, the chorionic cavity, the amniotic cavity, the embryo after the folding, the neural tube which is present, the pharyngeal arches which is present, developing heart, developing septum transversum, and these are the villus spaces. So here you can see the notocard, the neural tube, cardiogenic area, the oropharyngeal membrane, neuroenteric canal, the primitive streak, the cloacal membrane, allantois, and the connecting stalk. Also in this area, another thing which I have to show you over here, that this is the primitive yolk sac which is now present in the form of the cyst, and this is the definitive yolk sac which is called the umbilical vesicle. This one is the amniotic cavity, this one is the chorionic cavity, the splanchnic layer of extraembryonic mesoderm, the somatic layer of extraembryonic mesoderm, cytotrophoblast, syncytial trophoblast, intervillus space. So that's all about all this model. Thank you very much for your attention, and Allah Hafiz till the next class.